Good day, everyone. Here in front of you is Contillo, a BSN 1B student, and I am tasked here to discuss essential topics and lessons under our course Microbiology and Parasitology. So first, let me define you what is microbiology. So it is the study of microorganisms or a collection of organisms which cannot be seen by the naked eye but with the help or use of microscope. Now that I gave you a little definition of what's microbiology, since it's the study of microorganisms, I'll give you a question that would, I think, arouse the curiosity or your curiosity. So, are microorganisms beneficial to us? Or do we human beings derive benefits from these microorganisms? Well, yes. Microorganisms produce the food that we eat. Take for example, fermented dairy products like sour cream, yogurt, butter, fermented food like bread, pickles, or even alcoholic beverages. What amazed me is, the next thing that amazed me is that even the industrial corporations, scientists, they cultivate microorganisms, they study it and use it. They use microorganisms to produce some vitamins, enzymes, organic acids, and even other essential growth factors. So since microorganisms do help us, of course, if a thing has an advantage, it also has disadvantages. So another question that I will give is this: are microorganisms dangerous or harmful for us? Or do microorganisms bring, or is it detrimental to our health? Well, of course, yes. Microorganisms do bring harm unto us if they have the sheer force of numbers, or it could also be when they produce powerful toxins that affect or interfere our human body systems. Take for example, a bacteria that cause botulism and tetanus that produce toxins affects the nerve impulses. Like all other living organisms, microorganisms can also be classified as one, prokaryotes, and the other one is eukaryotes. So what are the difference and similarities of prokaryotes and eukaryotes? Having knowledge of this eukaryotic cell structure and prokaryotic cell structure will give us insight about their physiology. So now, I'll be discussing to you the prokaryotic cell structures and the eukaryotic cell structures. So first is the prokaryotes or prokaryotic cell. In the five kingdom of classification of organisms, prokaryotes constitute the kingdom Monera. So under the kingdom are the following: bacteria like the cyanobacteria, archaebacteria, or eubacteria. Prokaryotes do not envelope nucleus, or it has no true nucleus. Its ribosomes are a lot smaller than the eukaryotic cell. Also, there is no evidence that a prokaryotic cell has a mesotic structure like equatorial plate, aster, and another thing that prokaryotes do not contain is the organelles. Take for example mitochondria, Golgi bodies, lysosomes, or endoplasmic reticulum. Prokaryotes, its DNA is compressed into a nucleus-like body which is called the nucleoid, and it is not limited by a membrane. What are the structures of a prokaryotic cell? So here's a figure of a diagrammatic composite of a prokaryotic cell. First is the flagella. Flagella helps the bacteria to move. It propels the organism in rotary motion. Flagella is usually composed of a globular protein flagellin. So it is extremely long and thin, and it cannot be seen by a light microscope, but flagella can be visible through a use of an electron microscope. So it basically has three parts. First is the filament, which carries the flagellin. Next is the hook, which is composed of a different protein and lies along the proximal end of the filament. And lastly is the basal body, which anchors the filament to the cell membrane and cell wall. So the question here now, do all bacteria have the same form of flagellation? No. The monocrious bacteria has one single flagellum. The amphitrechous bacteria, or the amphitrich, has a flagellum at the end of each end of the cell. The lopotrichous bacteria has multiple flagella at the ends of the cell, while the peritrichous bacteria has a flagella distributed over the entire body of the cell. Next structure is the capsule. So the capsule provides an advantage to the bacteria. Why? The capsule protects the bacterium against environmental changes through creating barriers against drugs, antibodies, and other chemicals. Another one is the pili, or also known as the fimbriae. So pili is usually composed of a protein. So pili enables the cell to attach to other cells through a surface. Next is the cell wall, or the unique cell wall of a prokaryotic cell. So the cell wall of a prokaryotic cell contains a molecule called peptidoglycan. Peptidoglycan is a polysaccharide containing N-acetylmuramic acid, and another one is the N-acetylglucosamine with numerous chains of amino acids. Then here comes the cell membrane, or also known as the plasma membrane. Its phospholipid molecules are arranged into two parallel rows of phospholipid bilayer. So the inner and outer surface of the bilayer are composed of a polar head. So usually polar heads they contain phosphate groups and glycerol molecules, while on the interior surface, or if this is the phospholipid bilayer, so inside this phospholipid bilayer, or in between this, are the nonpolar tails that contains the hydrophobic fatty acids. So what is the role of the cell membrane? So cell membrane is selectively permeable. What does this mean? So there are certain substances which can penetrate through the membrane, while there are other substances which are not allowed to enter through the membrane. So it creates a barrier between the cytoplasm and the external environment. Now, the question is, how do substances enter into the prokaryotic cells or eukaryotic cells? So, in simple diffusion, the solute molecules move down, or it penetrates. If this is the plasma membrane, it penetrates through the plasma membrane. The solute molecules move down from a high concentration gradient area. From a high concentration gradient to an area of low concentration gradient. Meanwhile, in facilitated diffusion, as the name suggests itself, it also enters the plasma membrane from a high concentration gradient to an area of low concentration gradient, but actually, but the carrier proteins facilitates its transport to the plasma membrane. To add up some information, there is other type of diffusion. So it is called osmosis. So it differs from the simple diffusion and facilitated diffusion because it's the opposite, or it works the other way around. So the water molecules are selectively permeable, and it means that the water molecules move from a low concentration area to a high concentration area of solute molecules. There is a pressure that encourages the water movement from a concentration. This is called the osmotic pressure. Now, there are different types of water movement depending on the concentration, the solute concentration. First is the isotonic. So how does this work? Take for example, my hand is the cell membrane, and there are water molecules here. So the water molecules will stay at ease and will not move if a particular solute outside the cell is equivalent to the concentration inside this cell. Meanwhile, the concentration will become hypertonic if the solute concentration outside the cell is higher than inside the cell. So this water will move out of the cell because the concentration outside the cell is higher than what's in the inside. The last one is the hypotonic concentration. So how do this work? So in hypotonic concentration, the water will move into the prokaryotic cell because the outside of the cell, the solute is in a lower solution than the cytoplasm inside this cell. So that's how they differ from each other. Next structure inside the prokaryotic cell is the cytoplasm, or also known as the cytoplasmic area in a cell. This is the cytoplasm usually composed of 80% of it are water, carbohydrates, proteins, lipids, and other organic and inorganic compounds. So, cytoplasm really differs from the cytoplasm in the eukaryotic cell because the cytoplasm contains no organelle, no cytoskeleton of microtubules and microfilaments, and also it differs from its DNA, ribosomes, and other bodies. Since prokaryotic cells do not contain nucleus, however, in the cytoplasmic area, there is what we call the nucleoid. So in the nucleoid, packed into it are a single long chromosome in a form of a closed loop. So in the chromosome, it consists of a double-stranded DNA called the plasmids. The plasmids is used for genetic engineering, meaning it can be opened and some DNA fragment could be inserted into it, then it will be closed, and those new DNA fragments that was inserted into the plasmids could be inserted into another bacterial cell in order for that genes to express themselves in a form of a protein. Now, the site of protein synthesis is in the ribosomes. So ribosomes freely exist in all the areas of the cytoplasm. And the last one is the inclusion. So inclusion are various kinds of ultramicroscopic bodies. It may contain polysaccharides, lipids, sulfur, phosphate, and other materials which are needed for the biochemistry of a certain cell. So let's move on to another topic which talks about how do organisms reproduce and experience population growth. So first I will discuss to you about how do prokaryotic cells reproduce. So in prokaryotic cells, there is a process called binary fission. So it differs from how eukaryotic cells reproduce because in eukaryotic cells, the process is known as the mitosis or the cell division, and that's how eukaryotic cells experience population growth. So in binary fission, I'll explain it to you. I hope you understand. I'll help you get my point. So here is a diagram or an illustration of a binary fission, but I will also explain it to you using my hands. So take for example, this is a bacterial cell. This is a cell, a bacterial cell. So inside this cell lies the chromosomes. So here inside this cell lies the chromosome, which contains the double-stranded DNA called the plasmid, which I have discussed just earlier. So even before the binary fission begins, the chromosomes inside this cell has already duplicated itself. So just imagine the rolling circle mechanism taking place at this cell, the membrane of this cell. So once the chromosomes inside this cell duplicated, it attaches to the cell membrane. In the cell membrane, take for example, this is the cell membrane here. This is the cell membrane. The chromosome attaches here. Then the chromosomes grows here, and it separates. So if as it grows, the chromosomes separate. And then at this point, the bacterium contains two chromosomes in the region called nucleoid. What I have mentioned earlier, the chromosome is the chromosomes are usually found in the nucleoid part of the cytoplasmic area. Now, as the chromosomes separate, the cell wall or the cell membrane begins to form a transverse septum. So take for example, it separated, the two chromosomes separated. So it will now form a transverse septum. So it will start to be like this. Then, as the septum becomes complete, take for example, this is the complete process of, as transverse septum becomes completed, the two new daughter cells separate. So first, let's go back. The two as the chromosome grows into the cell membrane or cell wall, it separates. It starts to create a transverse septum. Now, as the septum becomes completed, two new daughter cells are being formed and become separated. So it will separate like this. Now, this is the magic. This is the amazing thing that takes place in a bacterial cell. From one cell, it created two new daughter cells. So from here, from this single cell, it produced two new daughter cells. Amazing, right?